The Northwestern University synthetic biologists have developed a low-cost, easy-to-use handheld device that can let users know within mere minutes if their water is safe to drink. The new device works by using powerful and programmable genetic networks which mimic electronic circuits to perform a range of logic functions. Among the DNA-based circuits, for example, the researchers engineered cell-free molecules into an analog-to-digital converter (ADC), a ubiquitous circuit type found in nearly all electronic devices. In the water quality device, the ADC circuit processes an analog input, that is contaminants, and generates a digital output, that is a visual signal to inform the user. The research was published in the journal Nature Chemical Biology. Equipped with a series of 8 small test tubes, the device glows green when it detects a contaminant. The number of tubes that glow depend upon how much contamination is present. If only one tube glows, then the water sample has a trace level of contamination. But if all 8 tubes glow, then the water is severely contaminated. In other words, the higher concentration of contamination leads to a higher signal. Northwestern's Julius B. Lux, who led the research, says that the tube with the lowest threshold will light up all the time. If all the tubes light up, then there is a big problem. Building circuits and programmable DNA computing opens up many possibilities for other types of smart diagnostics. The new system builds off work that Lux and his team published in Nature Biotechnology in July 2020. In that previous work, the team introduced ROSALIND, which could sense 17 different contaminants in a single drop of water. When the test detected a contaminant exceeding the US Environmental Protection Agency's standards, it either glowed green or not, to give a simple, easy-to-read positive or negative result. To develop ROSALIND, Lux and his team employed cell-free synthetic biology. With synthetic biology, researchers take molecular machinery, including DNA, RNA, and proteins, out of cells, and then reprogram that machinery to perform new tasks. At the time, Lux likened ROSALIND's inner workings to molecular taste buds. And now, in the new version dubbed ROSALIND 2.0, Lux and his team have added a molecular brain. Lux says that the initial platform was a biosensor which acted like a taste bud. Now they have added a genetic network that works like a brain. The biosensor detects contamination, but then the output of the biosensor feeds into the genetic network or circuit, which works like a brain to perform logic. Researchers freeze-dried the reprogrammed molecular brains to become self-stable and put them into test tubes. Adding a drop of water to each tube sets off a network of reactions and interactions, ultimately causing the freeze-dried pellet to glow in the presence of a contaminant. To test the new system, Lux and his team demonstrated that it could successfully detect concentration levels of zinc, an antibiotic, and an industrial metabolite. Giving the level of contamination rather than a simple positive or negative result is important for informing mitigation strategies. The researchers say that different contaminants at different levels require different strategies. If we have a low level of lead in our water, for example, then we might be able to tolerate it by flushing our water lines ahead of using them. But if we have high levels, then we need to stop drinking our water immediately and replace our water line. Ultimately, Lux and his team hope to empower individuals to test their own water on a regular basis. With inexpensive handheld devices like ROSALIND, that may soon become a reality.